Bioinformatics tools for analyzing pharmacogenomic data. Bioinformatics plays a crucial role in analyzing the large volumes of data generated in pharmacogenomics research. It involves the use of computational tools, algorithms, and databases to process, interpret, and extract meaningful information from genomic and pharmacogenomic data. Here are some commonly used bioinformatics tools for analyzing pharmacogenomic data. Alignment and variant calling tools. Alignment tools such as Bowtie, BWA, and STAR are used to map DNA sequencing reads to a reference genome. They align the short reads generated by sequencing platforms to the appropriate genomic location. Variant calling tools such as GATK, SAMtools, and Freebase identify genetic variants such as single nucleotide polymorphisms, insertions, deletions, and structural variations by comparing the aligned reads to the reference genome. Database resources. Databases provide a wealth of curated genomic and pharmacogenomic data that can be used for variant annotation, interpretation, and association studies. Resources like DBSNP, ClinVar, PharmGKB, and Cosmic contain information on genetic variants, their frequencies, functional annotations, and their associations with drug response or disease. Variant annotation tools such as ANNOVAR, SNPF, and VP, also known as Variant Effect Predictor, annotate genetic variants based on their functional impact. They provide information on the potential effects of variants on protein structure, gene function, regulatory elements, and known functional domains. Annotation tools help prioritize variants for further analysis and interpretation. Pathway analysis tools such as Ingenuity Pathway Analysis or IPA, Gene Set Enrichment Analysis known as GSEA, and Reactome enable the identification of biological pathways, gene networks, and molecular interactions associated with drug response or specific phenotypes. These tools help uncover underlying mechanisms and provide insights into the biological processes influenced by genetic variants. Pharmacogenomic prediction tools such as PMKB, Precision Medicine Knowledge Base, CPIC, Clinical Pharmacogenetics Implementation Consortium, and Precise PGX offer evidence-based guidelines and recommendations for drug dosing and treatment selection based on an individual's genetic profile. These tools integrate pharmacogenomic data, clinical evidence, and drug-specific information to guide therapeutic decisions. Statistical analysis tools such as PLINK, R, and SS enable the identification of genetic associations with drug response or disease outcomes. They facilitate genotype-phenotype correlation analysis, population stratification assessment, and statistical modeling to identify genetic markers associated with drug efficacy, toxicity, or treatment outcomes. Data visualization tools such as the Integrative Genomics Viewer (IGV), UCSC Genome Browser, and R packages, for example ggplot2, provide graphical representation of genomic and pharmacogenomic data. Visualization tools help researchers and clinicians interpret complex data sets, explore genetic variants, and identify patterns or trends. These bioinformatics tools, among others, aid in the analysis, interpretation, and translation of pharmacogenomic data into actionable insights. They enable researchers and healthcare professionals to identify clinically relevant genetic markers, understand their functional implications, and apply this knowledge to personalized drug therapy and improve patient outcomes. It is important to select the appropriate tools based on the specific analysis requirements, data type, and available computational resources.